Okay, great. So, how do I do this? Great. Okay, so I will present what we do in the lab, and that would be developing some lines that are responding differently to stress, and specifically during the juvenile period. So for this, I will go through different points. So I know that some of you do not work with rats, so I will present basically some basic stuff about the laboratory rats. Then I will present the stress system, a breeding procedure that we use in the lab, and some experiments that I did with those animals. So this is a table showing few parameters that I thought may be interesting to know the difference between human and rats. For example, the heart rate. So they have a heart rate that is much faster than us. They are smaller, but they have like 3 times higher heart rate. Concerning also the litter size, when they're born, they are between 6 and 12. I will mention this a bit later. When they are adults, and here I show a graph of the evolution of some animals that have been tested until 1.5 years. They go close to 600 grams, and then they kind of plateau to this maximal body weight. Their lifespan is about 3 years, but most of the time we keep them less because we need the brains before the 3 years. And in terms of development and weaning, the weaning date is after 3 weeks after birth. We do the weaning, so we remove them from the mother, and the sexual maturity is around 50 days old. Concerning the reproductive parameters, the females have an oestrus cycles of around 5 days, and the gestation is also about 3 weeks. So when we do the breeding procedures, we use 3 weeks for the gestation time and 3 weeks for the weaning procedure. This is a little timeline showing the first and early development of the animals from neonatal period, the 2 first weeks, the weaning after 3 weeks, the peri-adolescence, then the puberty around P35 to P50, 50 when they reach sexual maturity, then they were considered young adults, and in the lab we test them after P90 that we consider being adults. So I will just mention also 2 different points, is that during the 2 first weeks of life, they are in a period that we call the stress hyporesponsive period. It means that the development is still ongoing, and the development at that stage is using and is dependent on glucocorticoids. So the animals are developing a hypo-response to the glucocorticoids. So this is different in humans because this stage in humans is happening at the end of the gestation, and when the baby are born in humans, they are not hypo-responsive because the system is already fully developed. So this also means that the HP axis, the stress system, is still under development during the first weeks of life in the rats. Also something that is also the case in humans is that the hippocampus is still under development, and until adolescence it can be affected and the plasticity can be altered due to stress, which means that this period is specifically interesting in developmental studies and the development of psychopathologies. So here is just a little table that people try to do, comparing the age of the rats with the age of the human. And you can see that in the early aging and around the weaning, the aging of the rats is really slow compared to when they are adolescents and adults, and then it kind of reaches a plateau. So it's not linear. So this is, I think, the take-home message of this table is that the aging of the rats and the humans cannot be linearly compared, and it's not a linear relationship, and depending on the period of the development, they have a different correspondence. So now I will just present what we use to stress the rats during the juvenile period. So we call the juvenile period between P28 and P30, which are just before entering the puberty. So for this, we use one stressor that is illustrated. It's actually, we don't use a fox, but we use a molecule, the TMT, that is found in the feces of the fox. And this is innately inducing fear in the rats and the mice, because the fox is a predator of the mice. The second stress that we use is also environmental. It's an elevated platform. So we place the animals on a platform that is 1 meter high in a bright environment and in an open space. And this is a combination of environmental stress that induce innate fear in the animals. So what I want to say with this slide is that we stress the rats in an environmental way to induce fear, and we don't stress them physically with food shocks or other parameters for swim. So then I will describe the stress system, which is pretty important for us, and the HP axis. HP axis is just the hypothalamus pituitary adrenal axis, and is the axis that is involved in the stress response. So I will describe here a bit more in detail. So in the hypothalamus, we have this paraventricular nucleus, the PVN. This PVN is integrating different signals from the brain. And in case of stress or challenge responses, it induces the secretion of CRH, the corticotropin releasing hormone. These hormones will then act in the anterior pituitary and lead the secretion to another hormone that is the ACTH. The ACTH, the adrenocorticotropic hormone, will enter the bloodstream and circulate until the adrenal glands just above the kidneys and act on the cortex of the adrenal gland to secrete the glucocorticoids. So the glucocorticoids are the cortisol in human and the corticosterone in the rat, but here I will just use cort. And this is the end factor of the HP axis that will act in a lot of different parts of the organisms, in the muscles, in the liver, in the brain as well, to induce energy availability for response to stress, and as well as different behaviors. And importantly, this corticosterone is inducing the negative feedback. So it will act directly on the anterior pituitary as well as in the PVN to reduce the stress response. So we can see the stress response like here. So after a stress, the initiation of the stress, the pituitary is secreting the ACTH, and it will peak after 15 minutes. And in the same time, the increase in ACTH will lead to the increase in corticosterone that will peak after 30 minutes. The corticosterone will then act as a negative feedback and automatically reducing the stress response. And usually after 1 hour at the end of a stressor, we would expect the organism to go back in a basal level of corticosterone. And the other concept that we use in the lab to develop the lines is the stress habituation. And this concept is pretty easy, is that when an organism is subjected several times to a stressor, the first time it will induce a high response to stress. So for example, this could be the concentration of corticosterone, but the second day, the corticosterone is supposed to be a bit lower. It is habituating and adapting to the stress response. And here you can see two different extreme situations. In a case where an animal is not habituating to stress and always secreting a huge amount of corticosterone, and a rat that is habituating a lot to stress. And those habituation to stress and corticosterone response after stress have been used in the lab in order to develop 3 different lines of animals. So this is what we did. We took rats, males and females. We did the breeding in the laboratory. Then after the weaning, we put them in cages, 2 of them per house so 2 males together or 2 females together. And when they are just before puberty, in the juvenile period, we stress them in 3 different days. And so the stress that we use is the first day we do an open field that we use as a stress and also to assess the activity of the animals at juvenile period. Then we do an elevated platform, TMT, so the fox odor, also the elevated platform. And on the third day, we use the same stressor, but in another sequence in order to avoid predictability of the stressors. We also take some blood from the tails at P28 and P30, and we analyze the blood with the ELISA immunoassay, as you know, and we determine the concentration of corticosterone in the plasma. So this is what we obtain for the P28. All the animals have a huge corticosterone secretion in response to stress, but we were able to differentiate three kinds of animals: the high lines that do not habituate to stress and have the same corticosterone secretion after 3 days, an intermediate line that do show habituation but reasonably, and the low line that after 3 days of stress they already have a blunted response. And we could see the low and the high line as two extremes in the way they respond and they adapt to stress. So this is the basic statement of the development of our lines. And now I will show a bit what is happening during the breeding procedures of those lines. So we insemination, it will depend. After 3 weeks, then the pups are born. So we usually obtain between 6 and 14 pups, but in order to avoid body weight differences due to the size of the litter, we keep only between 8 and 10 animals in the litters, and we then wait 3 weeks to do the weaning. Then we pair out the animals of the same sex in the same cages, and we do not mix the different lines in different cages. And then we can do the cast protocol. So finally, the selection process for the next breeders is done like this. The high animals are the ones that secrete a high amount of corticosterone, and the low below 100 nanogram per milliliter. And this, we keep them, those animals. When they are adults, we again do the breeding between males and females of the same line. And we have been doing this for 12, so 11 generations in the lab. And here are the results of the corticosterone. And you can see that already from F1, the high lines were highly responding. And then the intermediate and the low line segregated in different corticosterone patterns. Even if you can see some variability between the generations, the trait is pretty stable, and we are pretty sure that we have a genetic selection for this adaptation to stress during juvenile period. So those animals are interesting mostly because they react differently to stress, and we want to analyze them when they're adults to see the behavior and also some metabolism and autonomic regulations due to the fact that they react differently to stress. So one important aspect is that the animals that I just presented before that are going through the cast and that are used as breeders for the next generation, they are not used for behavioral assessment. What we do is that with the generation and the pups of each generation, half of the animals go through the breeding procedure to be selected as next breeders, and the other half is kept for experimental testing. So this is important because all results about experimental animals with the lines have not been stressed during puberty, before puberty. So those animals we can test in different conditions. So I will just illustrate here a few of them that we used in the lab. For example, anxiety-like behaviors. So for this, we use the natural propensity of the animals, of the rats, to be scared of light and open fields. For example, here in the light dark box, the rats will be more comfortable in a dark environment than in a light environment. In an elevated plus maze, those two arms are closed, meaning that rats will spend most of the time in the closed arm and will enter sometimes in the open arms. And we can relate anxiety-like behavior with the amount of time that they spend in the open arms. And we say that one rat that spends a lot of time in an open arm is less anxious than one rat that stays all the time in the closed arms. And similarly, in an open field where we can have the activity, but also going more in the center and being more exposed to potential predators is also a sign of decreased anxiety. We also did some reaction to stress. So two different stress that we use in the lab are, for example, a restraint stress. So the rat is just inserted into a mesh-like restrainer, and it cannot move for between 15 and 30 minutes. And we take blood from the tail to see the corticosterone amount. And also the other one that is represented here by a bucket, we call it a novelty test. And basically we put the rats in a bucket, which is just a novel environment. So the rat cannot escape. It's not a really scary environment, but it's novel, so it's measuring the reaction to novelty. We also can do some memory and learning tests with the famous Morris water maze, but I will not present data of this since I don't have time. And an important behavior that we do in the lab and that was included in the MATRICS is the aggressive behavior that we measure in the resident intruder test. And in this test, all the experimental animals from the different lines are left in their home cages, and we insert a smaller rat that we call the intruder in the home cage of the resident, and we observe that the resident will defend his territory and will be aggressive toward the intruder. And we assess and we score the amount of time that the resident animal is spending attacking the intruder, and we can have a marker of aggressivity. We can also score different abnormal behaviors if one rat is attacking more when the intruder is in submissive posture or is biting vulnerable areas. So I will present here a few results that we obtained with the lines and that have been published last year. So first of all, during a restraint stress, we observed that the three lines mounted stress response after 30 minutes, but the low and the high lines, actually there is the linear difference between the three lines. The high are producing more cort, the intermediately, and the low have a low response to stress. And this is interesting because, as I said before, those animals have not been stressed during the juvenile period. So we have a constitutive difference in stress responsiveness between the lines without being exposed to stress during a juvenile period. Then with an elevated plus maze, we assess the anxiety-like behaviors, and we observe. So here we have the time the animals are spending in the different zones, and we see that the high line is spending more time in the closed arms and less time in the open arms than the low line. So this makes us think that the low lines are less anxious than the high lines. In this situation, you can see that the intermediate line seems to be similar to the high line. And actually the intermediate lines, depending on the generation, they kind of oscillate between the low and the high in terms of anxiety. So in average, they are intermediately anxious, but depending on the generation, there is this oscillating behavior. And in terms of aggressivity, as I mentioned before, here we saw clearly that the high lines were more aggressive, so doing more offensive behaviors. Also, the latency to attack was lower, and they were pathologically aggressive as well. I don't have the graph here, but they were also attacking more the abnormal, with abnormal patterns of biting in vulnerable areas in comparison to both the low and the inter animals. So for this situation, the high line seems to be pathologically aggressive and were of specific interest also in the context of MATRICS. And finally, I will describe little experiments that I did with those lines on the autonomic nervous system. So here we were targeting the autonomic nervous system because the sympathetic system and the parasympathetic through the vagus nerve are the major regulators of the heart rate. But in addition, the autonomic nervous system and the HP axis are also related. They are also cross-interact in different brain regions. The corticosterone and the glucocorticoid receptors are also present in the different central regions regulating the heart rate, and it made it a good potential target to test. So to do that, we were implanting the rats with fully implantable telemetric system with 2 electrodes, one above and one below the heart. And due to the electric signals of the heart, we are able to determine in live the heart rate of the animals during basal conditions, but also during behavioral experiments. So what we do to analyze those behavior is that we use specific software to determine all the spikes, so all the spikes of the heart rate, and by calculating the interval between two successive bits, we can determine what is called the RMSSD, the root mean square of the successive intervals, and this is a marker of the vagal tone. The vagal tone is the parasympathetic influence of the heart rate, and I'm sure you know it's just the system that is slowing down the heart rate. But more interestingly, it has been shown that resting values of vagal tone has been related to cardiac health, and also to the fact that an organism might be able to adapt flexibly to threat and to stress. And it is a good marker to see, it is the physiological flexibility and the response to stress through the heart. So in long term, studies have shown that low vagal tone, showing a low flexibility of a cardiac response, is related to cardiovascular disorders as well as psychopathologies. So more and more research are suggesting that the RMSSD might be a good diagnostic tool. And this is of interest because validating animal models with differences in vagal tone might also make the model more powerful in terms of pharmacological treatments as well. So what we did with those animals is to record the resting heart rate for several days. So here you can see dark phase, light phase in 2 different days. And really strikingly, what we observe is that the inter animals had a higher resting vagal tone than the low and the high lines. So actually, it's better to see the opposite, is that the 2 extremes in terms of stress responsiveness have a decreased vagal modulation, meaning that they are less flexible in the way they react and in the regulation of the cardiac health. This was related with a significant increase in resting heart rates for both extremes. So the link between the vagal tone and the heart rate is pretty easy. If we have a decrease in parasympathetic system, we have a decrease in what we call the heart rate break, so the heart rate is higher. But most interestingly, as I said before, this is an indication that in both the low and the high lines, there is a cardiac susceptibility and a decrease of cardiac health. So finally, I will just sum up few results from the 2 extreme lines that make us think that they could be good models of psychopathologies. For example, the low line, which has a blunted HP axis, as well as a low vagal modulation of heart rate, could be used as a model of PTSD, since low HP axis and low vagal modulation have been found also in PTSD subjects. We could also expect to see some susceptibility to stress and maybe susceptibility to develop PTSD. So in that sense, we are also trying to do some studies with fear conditioning and expecting to see if we can have a PTSD-like model with the low lines. And in terms of the high lines, what we observe is that they have hyperactive HP axis, an increased aggressiveness, as well as the lower vagal modulation. So this is also why we think that this line could be a good model of a conduct disorder, since conduct disorder have been shown to have an inadequate vagal modulation, as well as deficiencies in emotion regulations. And moreover, this could also maybe the stress of a model of resilience to stress that we would like to test further. And I would like just to show this U-shape that we already suggested in a review, that the HP axis activity in both extreme cases of a low or high activity has been linked with increased aggressiveness. And in this situation, we could say that kind of the low and high reaction to stress of the lines have been linked with the also decreased vagal modulation. So this is also something that we would like to suggest as a good model that, of course, needs further investigation, but in the link between aggressiveness and the vagal modulation.